As an engineer, I like numbers, tools, and measuring things, and there are still so many things in the brain that we still can't really measure, we can't quantify. So I'm trying to help neuroscientists by developing methods and software for them. So as I'm saying, brain is a fascinating structure, but we still don't know much about it. And as every other part of our body, even though it's amazing, it still gets affected by aging. So some people will age very well for a very long time, but for some people, it goes a bit worse and they get dementia, stroke, and other neurodegenerative disorders. So I'd like to talk today about not only about the aging of the brain, but also the methods that we're using to look at it. And then I would like to also show why it's important to be aware that these methods are not always that valid when we talk about the aging brain as for the healthy one. So if we want to differentiate between the healthy brain and the one with dementia, in ideal case, we would take the brain out, we would cut it into very thin slices and look under the microscope. But well, obviously, we can't do it on the living people. So then what we use is MRI, so it's magnetic resonance imaging. It's the non-invasive method to take a look inside of the brain. That method shows us the structure and the function of the brain. However, it also has some limitations, because what we actually see is just an image. Then when we want to take any kind of measurement, we need to use some assumptions, some mathematical and statistical modeling, which has some limitations I want to mention now. So basically in the brain, we can distinguish different geometric structures. There is cortex on outside, there's white matter in the inside, and then some subcortical structures closer here to the bottom. And there are many operations happening in the brain all over the time. And all the process information is being transported, being sent by the highways in the brain, which are white matter tracts, which are these bundles of neurons we can see here. However, we can't see them in the MRI in the very straightforward way, because neurons are tiny and our resolution of images is not good enough. So we use some kind of mathematical and physics tricks for that. And what we will do once we have the image divided into these kind of voxels, which are three-dimensional pixels, we would we would look how the water behaves in each of these voxels, and we would take the principal direction, the main direction of where the water moves, and then go point by point and try to track this water movement, because we assume that that's how the neurons are directed in these parts of the brain. So this is, again, this is an assumption. This is some kind of mathematical modeling, and we use the model of the directionality of water here. But, well, basically the main question would be why do we actually do that? First of all, for pretty pictures. So once we actually track each point by point, we get these lines. And this is a very pretty picture of the brain. In front there will be some eyes. Here's the back of the brain. So what we can see, there are lines. The ones going from top to bottom, they're in blue. The ones from front to back, they're in green. And the one from side to side, kind of from ear to ear, they're in red. So these colors are actually informative. But apart from that they're quite pretty, they're also very useful. So we can see here, for example, in the blue, we can see corticospinal tract going from the spine towards the cortex, so the top part of the brain. It's in blue. Then in red, we can see the this corpus callosum, so this is the tract that connects to hemispheres, and some other connections on the outside. And it's important to look at that because, for example, in the case of the neurosurgery, neurosurgeons are actually looking at how the tracts are being in the brain to make sure that they're not cutting through them when they're trying to resect the tumor. So this is just a model, but even in this paper from 10 years ago, we can see that on the top we have the modeled tract, on the bottom we see the tract dissected from the same brain. We can see that it's being quite accurate. However, in aging, there are some changes that are happening to the white matter, and these bright spots, which are called hyperintensities, which are types of lesions, happening naturally in aging. So to some extent, 90% of individuals after 60 years old will develop these. Obviously, they are much more prominent in dementia. They're also connected with triple risk of stroke. These come from the vascular origin normally, so these come from the problems with the vasculature. And then because that, we can think about like, we're talking about the highways in the brain. Now we can think about it as a kind of a flood going from the river, which is your vasculature in the brain. So because there is some change in this road that we try to assess, because there is different water fraction there, then we start to have a problem with estimating where the roads actually were. And this is important because not only in tumors we use that kind of tracking for surgery, but also in the diseases that are typical for aging. So, for example, in Parkinson's disease, one of the, it's not really therapy, but one of the ways that we try to help patients is implementing some kind of electrodes in a deep brain. So the surgeon would need to go all the way through the brain, somewhere here deep, to implement the electrode, which would work something like a pacemaker, to alleviate the symptoms. And here, for example, there's an example of publication. This is not connected to the hyperintensities, this is a bit different problem, but it says that on the left side, we have the first implantation result, that's in blue dots over there, which wasn't really accurate for some problems, because of some problems with this tractography, with this process of estimating tracts not being perfectly accurate. So then after half a year, they had to make a second surgery, and then they implemented the probes marked there as in red, which is obviously a hassle for patients, also might be dangerous if you actually cut through the brain, through the tracts. We know that because from like animal studies, we try to cut the tracts and see what happens. So, well, it's obviously the important problem. The question I had was why no one has done that before. So, when we have a big lesion due to stroke or to tumor, it's easy to look at that and see the symptoms this patient has. So, it's quite easy to say, okay, they have tumors, so they behave this way, they have these problems. Similar, so you can make an inference based on one subject, one patient. In case of multiple sclerosis, there are similar lesions, but they're due to different processes, and they are much more connected with disability. So we need to look at like 20, 30 subjects to say statistically it seems like that has an effect. These changes are subtle. As I said, most of us will have them at some point. So it's very hard to say what they actually do locally. That's why I'm doing it now when I have access to the biggest data set that probably was ever done for brain imaging. So, sorry. So normal study in neuroimaging would have 20, 30, 50, 60, up to 100 subjects. There are some big studies, like maybe few of them, which are using 1,000, 2,000 subjects. So there are big funded studies between different universities. And then UK Biobank, which is collecting a lot of data, not only brain imaging, it's also like behavioral data and everything else. We'll have 100,000 subjects. Currently we have 22,000. So in this cohort, in 5 years, we will get a big number of cases of Alzheimer, also ALS and other diseases that normally would be hard to spot, so we can make a follow-up and see what differentiates these patients from healthy subjects. But also that gives me a lot of data to work on and look at the tracts that are affected by hyperintensities and compare them with the ones that are not, because I have big enough data set to sample different cases from there. So hopefully I have shown you that we can make some pretty pictures of brain connections, that is being affected by aging. both the connections, but also the way how we make these pictures. And it's quite important to take that into account, especially in a clinical setting. So now when we have good enough data to fix it, I'm trying to do that. Thank you.